Hello everyone, welcome to MMBS111 Introductory Microbiology. This mini lecture focuses on section 8.1. It's an introduction to genetics and genes. There are 6 learning outcomes for this mini lecture. The first is define genome, gene, chromosome, genotype, and phenotype. 2, describe the basic structure of DNA, including all of the chemical groups and base pairings. 3, explain the replication process of DNA. 4, explain what the term semi-conservative replication means. 5, name the main enzymes and their function in DNA replication. And finally, compare and contrast the synthesis of leading and lagging strands in DNA replication. So, genetics is the study of inheritance or heredity, and it involves a number of different aspects. So it could be the study of transmission of biological properties like traits from parent to offspring. It could also study expression and variation in those traits. Some geneticists study structure and function of the genetic material. So what do those genes do? And then also the study of changes in genetic material. So here we're talking about mutations and how genetic material changes from one generation to the next. So there's quite a bit of vocabulary that's involved in genetics and understanding DNA and genes. The first that we're going to talk about here is genome, and the word genome means the sum total of genetic material of an organism. Maybe you've heard of the Human Genome Project, where we're studying all of the genetic material of human cells. Most of the genome exists in the form of chromosomes. However, some organisms like bacteria also have plasmids that contain genetic material, and eukaryotic organelles, as we learned, also have some organelles present that possess their own DNA. The genome of cells is composed entirely of DNA. So we're not talking about RNA here. We are talking about DNA when we're talking about cells. However, the genome of viruses can vary. Some viruses possess DNA and others possess RNA. So there are some differences between chromosomes in eukaryotic cells and bacteria. A chromosome is a cellular structure that is composed of neatly packaged DNA, and that DNA can be packaged in different ways depending on the organism. So in eukaryotic chromosomes, the DNA is wound tightly around proteins known as histones, and this helps to compact the DNA into those very tightly wound, sort of X-shaped structures. The eukaryotic chromosome is also located in the nucleus. It is linear and it varies in number. So some eukaryotic cells may have a few chromosomes, while others have very many, hundreds of chromosomes. The bacterial chromosome is also condensed and packed into its more compact shape, but it doesn't use histones. They are proteins that are similar to histones, but not exactly the same. The bacterial chromosome is mostly single. So most bacterial cells have one chromosome, and it tends to be circular. However, there are some bacteria that have multiple circular chromosomes, and there are some prokaryotes that also have linear chromosomes, so not all are circular. A gene is a segment of DNA that contains code or instructions for a group of related proteins or RNAs. And there are three categories of genes. Some genes are structural, and that means that the instructions for the gene code for proteins. Some genes code for RNA machinery that is used in protein production. And then our final group of genes are regulatory genes, and these genes control the expression of other genes. And when we're talking about genetics and heredity, often you're going to encounter the terms genotype and phenotype, and it's really important to understand the distinction between these two terms. A genotype is the sum of all the genes. So it is the organism's very distinctive genetic makeup. What genes does it have? What does its DNA or its genetic material look like? The phenotype is the expression of the genotype. This is the organism's traits. For example, if an organism, a bacterium, might have the gene for a flagellum, the phenotype then is the expression of that gene or the presence of a flagellum. So while the genotype is the genetic material and what genes are possessed, the phenotype is the appearance or the expression of those genes. So DNA is composed of some very basic structures. We looked at these in chapter 1. There are 3 components of a nucleotide, and DNA is made up of a sequence or a chain of nucleotides that are bonded together and repeated. So we have a phosphate group, the deoxyribose sugar, and then a nitrogenous base, and this is what makes up the basic structure of a nucleotide. These nucleotides then bond to each other and become the backbone of each DNA strand. If you look at the diagram there on the right, you can see how these nucleotides sort of bond together. Each sugar attaches in a repetitive pattern to 2 phosphates. One phosphate is going to attach to the 5' carbon of that deoxyribose sugar, and one phosphate is going to attach to the 3' carbon of that deoxyribose sugar. If you take organic chemistry or biochemistry, you'll learn a little bit more about this, but when we're talking about structures that have a ring of carbons, those carbons are numbered, and that helps us to identify where bonds are occurring. So the number 1 carbon is the carbon that is bound to the nitrogenous base, and then you proceed around the ring in a clockwise direction. So that's how we get to the 3' and the 5' carbon, is by counting the carbons in the ring going clockwise. Nitrogenous bases attach along the strand, as I said before, at that number 1 carbon of the deoxyribose sugar. We have 2 types of bases. They are called purines and pyrimidines, and it's based on their structure. And those bases then are joined together in the 2 strands by hydrogen bonds, so that it can easily be unzipped or separated one strand from the other, and this helps to enable replication and transcription in protein synthesis, so that we can have one template strand that is unbonded that we can use to make an RNA or another copy of a DNA. Hydrogen bonds tend to be fairly weak, so they're easy to break and separate, which is why we have those hydrogen bonds between the bases. The bases pair in very specific ways. So adenine is always paired to thymine, and guanine is always paired to cytosine. So if you know what the base sequence is for one strand, you can determine the base sequence of the other strand because these pairings are exact. They don't pair with any other bases. The DNA double helix is said to be anti-parallel, and this means that one side of the helix runs in the opposite direction of the other. So the order of the bonds between carbon on deoxyribose and the phosphate is used to help us keep track of the direction. So one side runs from the 5' carbon to the 3' carbon, and the other side runs in the opposite direction from 3' to 5'. And this is really important when we start looking at DNA replication and protein synthesis, what strand is used as the template in those processes. So here we have a very simplified example of DNA replication. DNA replication is said to be semi-conservative, and this means that one strand of the original molecule is used as the template to make the new strand. So we don't make a brand new strand with two brand new sides. Each strand at the end will have an old template strand and a new strand. So we get two exact copies. This helps to ensure that the DNA is replicated correctly and appropriately, so that we're not trying to make something brand new without a template or a guide. This model here shows how DNA replication takes place, and we have a number of enzymes that are involved in this process. So you can see we're going to start over here on the left. We have DNA helicase, or just helicase. Helicase is an enzyme that helps to unzip the DNA molecule and separate it into two single strands. This is really important. It is responsible for breaking those hydrogen bonds between the two bases, and then it opens up sections of the DNA so that new bases can be added and two new strands can be formed. We also have single-stranded binding proteins that attach to each strand, and these proteins help to prevent the DNA from recombining and joining back together once it's been separated by the helicase enzyme. So there are two strands, I guess, when we're talking about DNA. We have a leading strand and a lagging strand, and this has to do with the orientation of the nucleotides on each strand. DNA polymerase III is the enzyme that is going to add the bases to that template strand to make the new molecule. However, DNA polymerase III can only work in one direction. It can only go from 5' to 3'. So the leading strand is the strand that is the opposite orientation of 5' to 3', and that is the 3' to 5' strand. So that's the leading strand, and DNA polymerase can add bases continuously on that strand without interruption. On the lagging strand, we have a little bit of a problem, and that's because DNA polymerase III can only add bases in one direction. And since the lagging strand is the same orientation as the direction that the bases need to be added, it has to add bases in little segments, and we call those segments Okazaki fragments. And each fragment starts with a primer. So DNA primase is going to add an RNA primer onto the lagging strand. And then DNA polymerase III will find that primer, and it will begin working in the 5' to 3' direction to add new nucleotides to the lagging strand until it meets the primer of another fragment. Once it reaches the primer of another fragment, then it's going to hop off the lagging strand and it's going to find another primer that does not have nucleotides added. And it will continue to do that all along the lagging strand. DNA polymerase I is going to come in and it's going to remove those RNA primers and it's going to replace them with DNA nucleotides. So those will get replaced. The RNA nucleotides will get replaced with DNA nucleotides. And then DNA ligase will come in after the primers have been replaced and join all of those fragments so that it becomes one continuous strand. Okay, so these are the key enzymes that are involved in DNA replication, and make sure that you know what they are and that you understand what they do. So that's the end of mini lecture 8.1. There are several questions here that you should go through and make sure that you understand as far as the surface content from the lecture. Make sure that you can answer these appropriately, and also go back to the learning objectives and make sure that you understand those as well. So here's a critical thinking question. Here's an example of a question that you might see on the exam. What would the new strand of DNA look like if this is the template strand? So if you were presented with this template strand, what would the complementary strand of DNA look like? In what direction would replication take place? So think about DNA polymerase III. What direction does it work in? Is this segment that you see here the leading strand of DNA, or is it the lagging strand? These are some things that you need to think about and be able to do so that you can be successful on the exam. So that's the end of 8.1. Hopefully this video was helpful.